Good morning. Today's topic is transcription. So in transcription, from DNA, RNA is formed. DNA is double-stranded, but both the strands will not act as template. Template means the strand from which RNA is made. Only one strand of DNA will act as template. Then the question is why RNA cannot be formed from both the strands of DNA? See, DNA is double-stranded, so this strand is complementary to this one. So if RNA is formed from this strand and again RNA is formed from this strand, so these two strands of RNA will become complementary to each other and a double-stranded RNA will be formed. Now problem is that double-stranded problem does not make any protein. Second point, anyhow if it is stopped that these two do not become double-stranded, then what will happen? This is complementary to this one. That means the sequence is different of the nucleotides. So the sequence is different, so different proteins will be formed from the same gene. So two different proteins from the same gene will be formed, which will be very complex. It will make the machinery very complexity will develop. Therefore only one strand acts as a template for the formation of RNA. Now second point is that which strand will act as a template. So this is decided by the promoter and the terminator. Before that, for the formation of RNA, enzyme RNA polymerase is required. The name of this enzyme is DNA dependent RNA polymerase because this RNA polymerase is dependent on DNA. See, this is the transcription unit. Transcription unit has a promoter, structural gene and terminator. So DNA, this is strand 3' to 5' and this strand 5' to 3'. So this promoter at the promoter RNA polymerase will attach, and we know that the polymerase reaction, the formation of the RNA strand will be from 5' to 3'. That means it will attach to the 3' to 5' of the DNA. That means this will be the template strand and this one will be the non-template strand. Now this non-template strand is also known as coding strand. How is it that it is known as coding strand? See, this is DNA double-stranded 3' 5' and 5' 3'. Now promoter will attach here, and now RNA polymerase, with the help of RNA polymerase, from 5' to 3' RNA will be formed. So suppose the sequence is ATCCCGGG. So as a double-stranded DNA, so second strand, the complementary strand will have TA, GGGCC. This is clear. Now from 5' to 3' RNA will start at the promoter region with the help of RNA polymerase enzyme. So as it is RNA, so there will be no thymine, it will be uracil. So the complementary will be U, then A, and then GGGCCC. Now if you accept this U and T, why? Because this is RNA, so in RNA thymine is not, uracil is there. So except this T and U, you can see the strand of DNA and the strand of RNA formed, and the non-template strand of DNA has the same sequence. You see A, G, 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 C, C, C. So this non-template is known as coding strand because this is the codes are same here. You can see for the RNA strand and the DNA strand. Further explanation will be done in the next short video.